Okay, we have 20 people now, and I think we will have more people in the next few minutes. I think. So, Jose, I think we can get started, right?
Sounds good.
Okay, welcome everyone to our bioinformatics and genomics seminar today. And this seminar is the last seminar for this year. And we're glad to invite Dr. Ahmed Mahfouz from Leiden University of Technology to talk about his genomics biology papers. As we know that the single cell RNA-seq analysis, the most time-consuming part is the cell class annotation. And then I think he will introduce a little bit more about different methods to do the cell cluster annotation and talk about his interesting research. For the next year, we have the first speakers on January 12th. And if you miss any previous seminars, our previous seminars were recorded in YouTube. So you are feel free to come here to watch the YouTube video here. With that, I will turn it over to Dr. Jose Maintains from BCH, who will introduce our speaker today. Thank you, Jose.
Yeah, thank you very much, Liang, and also Shira for the opportunity. And I'd like to spend a couple minutes going over what Ahmed Mahfouz has done over the past few years and kind of how he's arrived at this current interest in single-cell RNA sequencing. And Ahmed, maybe while I introduce, you can start projecting your slides and make sure that everything looks okay. So Ahmed started off in his scientific career in Egypt, and then proceeded on to his PhD work in Delft University of Technology in the Netherlands. And from there, he's also been involved with a variety of quite collaborative efforts, which I think speaks to the way that he approaches science, and something that we also appreciate here. Touching on a variety of different projects, including some with the Allen Brain Institute, and in collaborations with them, I think he, as well as many other of us here, have become quite captivated by the power of studying single cells. And so a lot of the work that you might be familiar with from Ahmed's recent group, which he started at Leiden University, also in the Netherlands, have really focused on trying to get a good grip on what is the right way of approaching our single cell analyses. But within that, there's a variety of different biological questions that his group also is interested in addressing, including the ever kind of pressing existential question of what is a cell, right? And what is a cell type? What is a cell subset? What is a cell state? And in particular with kind of gene regulation, and I think you might also share some new and ongoing efforts on eQTL loci and how he's integrating that into single cell data and our understanding there. So today, I think he's going to focus mostly on some of their recent work in using new cell identification algorithms to help us classify cell types in single cell RNA sequencing data. And with that, I'll turn it over to Ahmed. And please, if you have any questions, you can drop them in the chat, and then maybe we can get to them towards the end, unless Ahmed, you'd like to take questions during, but completely up to you, however you want to field that.
Yeah, if you want to interrupt me in the middle, please do so. I don't have an eye on the chat, so please, if there's anything there, just
sounds good. We'll monitor during, and if there's anything pressing, we can ask. But if not, please take it away.
Perfect. Thanks a lot. Thanks a lot for the invite. I really appreciate the opportunity to share some of the work that we're doing with you. So I'll just start my talk. So the main interest of the lab now is that we are very much interested in genetic variation. And basically what has been happening over the past few years is that these GWAS studies or exome sequencing studies have been very successful at identifying many variants that are associated with a wide variety of diseases. But that's in general a bit problematic for people who want to study the implication of these variants or want to move forward to develop therapeutics and so on. because most of these variants have very, very small effects if you're talking about a complex disease like Alzheimer's or so. And getting like a good understanding of the effect of these variants is pretty difficult. So the approach we take is a functional genomic approach. So many other people do that. And in the past, we've been focusing on these transcriptome atlases, like the data from the Allen Human Brain Atlas, where you map the expression of a certain gene across time, across development, or across different brain regions. And we have a lot of examples to show that this can actually give you a lot of information about the function of a specific gene or a group of genes in a specific brain area or a certain time of development. And of course, since the emergence of single cell data, this gives you a new dimension, right, where you can now go down to a single cell level and perhaps focus on which population is targeted or mostly affected by a certain set of mutations. So one of the things that are becoming one of the things that's becoming really clear and important here is that we kind of need to get a good grip of what is a cell, what is a cell identity. And I'm showing here like an example of why this is relevant. So this is a recent preprint from the Allen Institute where they compared basically cell types across different species. So you have human, marmoset, and the mouse. And what you can see here is that, for instance, looking at different types of interneurons, you can look at conserved markers across different species. And these are human-specific markers, marmoset-specific markers, or mouse-specific markers. And this is really important, right? Because if you're studying a specific human mutation, you create a mouse model for it. You would like this to be as representative as possible. And on the right here, this is just a graphic showing that there are, different species-specific expression patterns. And you would think these might not be very interesting things, but like if you're talking about one of the genes that are clearly different between mouse and human is serotonin receptor, which is targeted by a lot of drugs aimed at depression, for instance. So it is really important to actually know how genes and cell types differ across species. And for that, we need a good definition of a cell identity. So let's look at how cells are identified now in single cell data. So we started from a count matrix on the left here, where you have for each cell the expression of all the genes that you can measure. And what you end up with at the very end, so here I have an example from immune cells, is an annotation for cell clusters, basically. So that tells you that all these cells are B cells and so on. So how do we do that? Well, the typical approach is that you take the data, you cluster it into groups of cells, and then you start to overlay known markers that you know about the cell. So for instance, CD3, CD4, then that would tell you that this group over here are the CD4 T cells and so on. Yeah. And this works really well. I think that's the kind of standard with all the single cell studies. I'm shamelessly plugging in here a tool. This was released on bioRxiv yesterday. It's called Cytoexplore Transcriptomics that would allow you actually to do this in a very interactive way. And because of a trick that we do there in these low dimensional embeddings of the data, this allows you to interact with millions of data points, right? Which is usually a bit limiting. So if you rely on most of these visualization tools, they do downsample the cells to a manageable size. What we can do here is that we can actually deal with millions of data points. Do the typical analysis steps. You can manually select groups of points, do differential expression on the fly, overlay markers, and so on. So please have a look at it if this is the type of work that you're interested in. But okay, that's all very nice, but it also has a lot of problems. So it's time consuming. If you have done cell type annotation in single cell RNA-seq data, you would know this. It's really a painful step of trying to figure out what are these populations, especially in complex tissues like the brain. It's not reproducible, and I'm not talking even between different groups, even within the same group. If you ask different people to annotate cells, you will end up with different results at the end. And that has to do with the fact of how we perceive, for instance, marker expression and which markers do we rely on and so on. And of course, it's subjective at the end. Huh? So we can, even based on the same data, we can disagree about the annotations of specific populations. So can we automate this step? Huh? So to solve these issues. And if we do clustering, we are basically interested in like the distinction between these different groups, but we can also rely on classifiers or supervised methods that can describe these boundaries between the different classes. So the way this is done typically in any machine learning setting is that you have some training data. So these can be annotated cells, so they can come from a previous study or from an atlas that is available. Perhaps you also have some knowledge about marker genes. So you know that, for instance, CD4 T cells have to express CD4. And then once you have a new data set that's unannotated, so we haven't analyzed this data yet, you can run it through the classifier and you can basically get annotated cells automatically. And this would solve a lot of problems with the timing, subjectivity, and reproducibility. You might also be interested in flagging unknown cells or novel cell populations. So these are populations that you have not seen during the training of your classifier. And this is especially important and interesting if you are working with diseased samples. So where you would expect populations to differ from the training data, which probably would come from a healthy or undiseased data. Okay, so we actually came up with this idea, I think, 1.5 years ago, where we thought, okay, there are already a lot of classifiers that have been developed. Our experience from working on mass cytometry data, also with classification, is that these classifiers in general tend to be a bit more complex than they need to be. So what we did is that we made a list of all the classifiers that were available back then, and you can see them over here, scattered. This scattering is not random, so we group them also based on the underlying method. So all these classifiers here rely on a random forest, for instance. These are both based on a support vector machine and so on. What we did is that we also added 6 off-the-shelf classifiers. So for instance, we add an SBM classifier just from scikit-learn. We had one with a rejection option. This is just a basic random forest classifier and so on. So just to see also if the additional tweaks or adaptation that people add to the methods do help them in classification or not. So I'm gonna go through a set of experiments that we did in this benchmark study. I'm not gonna cover everything in the paper, but please, if you have any questions about it, do let me know. So the first experiment we did is an intra-data set evaluation. So basically we take one data set and we go through a classical machine learning evaluation using cross-validation. So we divide the data set into 5 folds. We use 1 fold for training, 4 folds for training, 1 for testing, and so on. We evaluate the performance using the median F1 score, just to summarize the precision and recall. And we also report the percentage of unlabeled cells. So if the classifier actually rejects cells or flags them as unknown or unlabeled, we report this percentage. And I'll show you in a bit why we do that. Of course, before I go through these results, that's a very artificial situation, right? So it's very unlikely that you're actually interested in this performance. Because it's on the same data set that you already have where you have the labels. The reason we do this experiment is to basically have kind of a common ground for all the tools that makes it a bit easier to evaluate the performance. Okay, I'll start explaining how the results are laid out. So here are all the classifiers that we have tested. On the columns are the different data sets. I start here by showing 5 pancreatic data sets. These are relatively easy data sets. They've been used a lot in all the papers. Most of the single cell papers, no matter what task they're looking at, they look at pancreatic data. And while this is convenient, I think this also shows you that it's really a very easy task. All the classifiers perform well on this data. Pancreatic data, if you don't know, have relatively a few number of cells, and they are quite distinct. So it's not surprising that most of the classifiers perform well on the data. What you see at the bottom here are classifiers that require prior knowledge in the form of marker genes, for instance. We did not test them on pancreatic data because most of them do not provide markers for pancreatic data. The vast majority provide markers for PBMC data, for instance, which already tells you one of the limitations of these classifiers is that to use them on a system, you need to already know a little bit about the markers that you need. The self-bench data is really a small data set of cell lines that are profiled with 2 different protocols, 10x and CellSec2. And again, this data is really very easy to classify. To be honest, the only reason it's in the paper is the reviewer was interested in that, so we added, but it's really a very easy data set. I told you we should not only look at the median F1 score, you should also look at the percentage of unlabeled cells, because for instance, if you look here at cell blast, for one of the cell bench data, it has a high performance and median F1 of 0.99, which is super high, but at the same time, it's rejecting close to 70% of the cells. And that makes the task of the classifier, of course, a lot easier, right? You will get a 1 of a median F1 score if you reject everything. So that's why we always look also at the percentage of unlabeled. Okay, so moving to the Allen mouse brain data. So this is a more challenging task. So we have data from the mouse cortex, and the data, it's one data set, but it's annotated at 3 different levels. So at the very easy level, so the first column here, you only have 3 classes. So you have non-neuronal cells, excitatory neurons, inhibitory neurons. And that's again, a very easy task, and what you see is that all the classifiers get described, right? It's not a challenging situation. At the second annotation level, we already go to 16 classes, and there you see that some of the classifiers already start to suffer, like the k-nearest neighbor, for instance, is already not doing very well on this data, while most of the other classifiers can still handle this. If we go to the deepest level that we have, that has 92 labels, then it becomes a relatively challenging task. And here you see that the performance of all the methods, no matter what, drops, right? Except for at the top, SVM with a rejection option still stays high. But as I said, if you look at the right, it is rejecting close to 20% of the cells. So it's also making its life easy by rejecting cells that are likely difficult to classify. Yeah. I want to point one thing here. So SCVI, which is based on a variational autoencoder, seems to do very well unless we go to these very deep annotations. And I don't think this is really a problem for SCVI itself. It's a deep learning model, so you really need a few examples to learn from. And at this deep annotation level, a lot of cell populations are actually very, very small. So that's something to keep in mind if you want to use these methods for data that has very rare populations. One thing to note here is that we include an SVM classifier with and without a rejection option. And you see that the one with a rejection option is doing better. But as I said, it's also rejecting 20% of the cells. So there is kind of a trade-off between how much do you want to reject and how high do you want your performance to be, right? So I think this differs also per application, where in some applications you really don't mind, you just want to get an annotation for all the cells, accepting few mistakes, while in other applications you really want to make sure that all the annotations are correct, and you will sacrifice few cells that end up not being annotated at the end. For PBMC data, so we included 2 data sets. So they are both coming from 10x. So the one on the right side, this is a large data set of close to 70,000 cells, annotated as you would annotate most of the single cell data. The one on the left is a smaller data set, but it's sorted population. So you fact sort different lineages of the immune system, and then you do single cell RNA-seq. So you're also a bit more sure about the identity of the cells. So what you see here is that the performance of most classifiers is also quite okay. I think we here had 11 populations to classify. One thing to note is that, again, for these classifiers that require having markers to do the classification task, they do not cover all the populations that were included. Although, as I said, the data is annotated at 11 populations, which is not very deep for PBMC data. Still, these classifiers like scINA, for instance, does perfect on this, but it's only classifying 3 classes, yeah, out of the 11, which is a much easier task, of course. So again, I think this is quite a problem for these classifiers that require markers, known markers. I will not discuss this further here, but in the paper, you would also see that coming with known markers from literature might be a bit unfair, right? Because the single cell data has very specific noise characteristics, for instance. So we also tried to define markers based on the data itself using differential expression, and then use these markers for the classifiers. Again, this is very tricky. And if you look at the overlap between the markers already implemented or included in these methods, you see very little overlap actually in between them for the same cell population. So to me, this points to a bit of a limitation of going in that direction. I think it's a much better and much more practical approach to rely on classifiers that would pick the signal directly from the data instead of providing it by the user. One thing I didn't tell you, so this is an overview of what I've been saying, is that these classifiers over here are ranked based on the median expression across all these experiments. And what you see is that at the top 5, we have the SVM rejection, which is an off-the-shelf classifier with a rejection option, a CPRID that's based on SVM, and SVM, again, that's from scikit-learn, and single-cell-net and actin. So we were a bit surprised actually to have 3 linear classifiers at the top of this list. One explanation for this is that the annotation itself comes from human annotations. So the authors of these data sets plotted markers and then gave annotations, which would also drive these linear separations between the classes, because if people rely on binary markers that are either on or off, then this would fit a linear classifier pretty well. But nevertheless, it's interesting to see that at the top of this list, very simple off-the-shelf classifiers that we really didn't tweak at all are outperforming classifiers that are specifically designed for single-cell data. I'm going to skip over this one. So the second experiment we did is to look at enter dataset evaluations. And this is a bit of a more realistic scenario where you have one dataset that you train your classifier on, and then you get another dataset. Assuming that you don't know the labels, you want to use your trained classifier to label this dataset automatically. Of course, in this case, we use data where we have the labels to be able to test. But it's in general a more challenging task than what I showed you earlier. So for this, we did a couple of experiments. I want to share one with you today where we looked at this paper. It wasn't still on bioRxiv back then, but they basically took samples from human and mouse cell lines, human PBMCs and mouse cortex nuclei, and they had 2 replicates of the same sample. And then they measured it with a wide range of single-cell RNA-seq technologies. And this is pretty nice, actually, if you're interested in comparing these protocols, but it's also good for us to have a look in terms of classification performance. So what we did over here is that we took one data set. So in this case, the 10x version 2 chemistry, and as our training data. And then as a test data, we use all the other protocols. So that can give us an idea on how can we, what is the performance if we train on one protocol, test on another one. What we can also do is to, the last column over here, is to train on the 10x v2 replicate 1 and test on 10x v2 replicate 2. Yeah, and that would tell us something about the replicability of the method itself, little about the classifier, because this task is relatively easier. These are 2 replicates of the same sample, 2 biological replicates. Of course, this is one combination, but we can also do this for all the possible combinations. So we can now take 10x version 3 as the training and then test on all the other protocols and so on. Keep in mind, for 10x v3, there was only 1 replicate. So that's why we don't have results on the replicability. Okay, I'm gonna point out to a few things. So we can also summarize the performance per classifier. And again, we rank them based on the median F1 across all the experiments. One thing to show here is that, for instance, SVM rejection is not the best one in this case. But if you look at the results, this is mainly driven by low performance on the SmartSeq2 data and the CellSeq2 data. For the rest, I think it's quite okay. Keep in mind that these are the only well-based data sets. The rest are coming from droplets. You can also summarize the performance per data set, and this really gives you nice information. So one example here is that the correspondence between 10x v3 and v2 chemistries is pretty high. The other thing is that SmartSeq2 seems to be more distinct from the other protocols And that's evident by the low performance of most classifiers on this data. So you might want to take that into account if you're merging multiple data sets that include SmartSeq plus other droplet-based methods. One thing that you see over here in terms of replicability, that for CellSeq2, this replicability is a bit low. And so you see that most of the classifiers actually have low performance when you go between the two replicates of the same technology. That's something that was also noted in the original paper, by the way. Okay, the third experiment I want to talk about is about rejection. So as I said, it's important if we are to have a good classifier that we can reject cell populations that were not seen during training. And that would make sure that the classifier performance is high, but that we can also flag cells that might be very interesting, right? Especially if we look at disease situations. A very common experiment to do here is to create these artificial situations where I basically train on a PBMC data and test on a pancreatic data. And in general, the classifier should reject everything. So these values should be 100%. You see that some classifiers are doing this okay. Some are actually suffering a lot. So even for SVM rejection, if you look here for the mouse data, if we train on pancreatic data and test on brain data, the classifier is still giving you kind of confident estimates for 3/4 of the data set, which is not great. But a bit more of a realistic scenario is that one cell population is missing actually from the data, so not so radical. So what we did here is that we trained classifier on a PBMC data, blood immune data, and then we removed one cell population. And we removed, for instance, T cells, and then we removed only CD4 T cells in another scenario. And then the third scenario, we removed the subpopulation of CD4 T cells. And what you see is that this is a lot more difficult, right? So most classifiers actually don't get this close to 100%. And we also see something a bit more counterintuitive a little bit, is that rejecting T cells in general is actually more difficult than rejecting a very specific population. And this has to do with the similarities between these cells. So for the classifier, these cells actually are quite distinct from all these subpopulations, while the T cells, they will end up being very similar to two subpopulations of CD8 T cells, for instance, if you remove the CD4. So this is a bit counterintuitive to get, but in general, this points out to the fact that rejecting cells is actually a difficult task. It's not very trivial. Most of these methods calculate some sort of a posterior probability of the classifier, and then they use that to reject cells. So if you have a posterior of lower than, I don't know, 70%, then these cells are rejected. But if you plot these posteriors for the cells that are actually truly classified or misclassified, you see that there is actually no distinction. So it's not a good criteria to use these posteriors for rejection bluntly. Okay, this is part of our summary figure where we basically show that, okay, across all the inter-data set experiments, the rejection, also the timing with respect to different numbers of cells or features or deep annotations, we can summarize our results into, good or intermediate and poor classifiers. Again, I think our conclusion from this is that relatively simple classifiers actually work quite well. Again, this is a bit surprising to us that these off-the-shelf methods are actually doing quite okay in terms of classifications. So I think this at least our hope is that this helps people who are interested in classifiers to choose what's the best option, but also for people who are developing new methods to take this into account. So to summarize a bit this part of the talk, again, these simple off-the-shelf classifiers do seem to outperform dedicated single-cell classifiers. And again, if you're interested in this, there's also a preprint from Fabian Theis's lab that basically comes to the same conclusion, although they did more extensive cross-data set experiments. So I highly recommend looking at it. But again, the message is the same at the end, that simpler classifiers are actually okay for the task. Adding these prior knowledge in terms of markers is actually not helping. It's more limiting because you're limited to what you know. And there's a very wide variability between these markers. So yeah, at the moment, I actually don't see the value of doing this. Perhaps this will change in the future, but at the moment, I don't think this is beneficial to do. Rejection is very important, but it's also very difficult to implement. To make it a bit more accessible to people to have a look at our results, there's a SnakeMake pipeline of this benchmark. So if you want to test a new classifier or you want to add a new data set to evaluate these methods, you can easily do that. So please have a look at it. All the data that we have used in the benchmark are also available. So that's also a nice resource if you're interested. Okay. For the next part of the talk, I actually want to talk about the applicability of these classifiers in a certain setting. So we are very interested, as Jose said, in EQTL analysis. So for those who don't know what an EQTL is, we basically try to identify associations between genetic variants, SNPs, and gene expression. And this is typically done by basically looking at a large data set where you have SNP arrays, you have gene expression, and then you try for a specific gene to see if a specific SNP has a correlation based on the different genotypes. EQTLs are also cell type specific, so it will be really good if we can identify these EQTLs that are present in one cell type and not another one, because that can help you also use these for targeting, drug targeting, for instance, downstream. So the way this is done, you have a cohort, you take genotypes, you also measure single cell RNA-seq. We're mainly talking about PBMC in this setting, but I think this also generalizes to other tissues as well. One problem here is that to get to enough statistical power to identify these associations, you do need a large number of individuals. So of course, this means that we need to go to cohort studies, and that eventually means that we need to consider multiple cohorts at the same time. So that's why this single cell EQTL consortium was formed. So this is led by Monique van der Weijst and Lude Franke from Groningen in the Netherlands, but with partners from all over the world. So please visit the website, have a look, and if you're interested in joining or in contributing data or joining discussions on method development and so on, please reach out to the consortium members. There is a white paper describing the consortium aim and goal on eLife, so please check it as well. One specific problem that we are interested in is how to guarantee the consistency of cell identities. So you can imagine if we're interested in EQTLs that are specific to CD4 T cells, we need to make sure that CD4 T cells identified in Groningen are the same as CD4 T cells identified in Joseph Powell's lab in Sydney. So one thing is that we can put all the data in one place, do the annotation, but it's very impractical. First of all, it's a huge amount of data. The second point is that not all the data is available at the same moment, right? This is eventually a growing cohort that will keep adding data. And one thing you want to be sure of is that whatever we find now will not change if we add more data next year, for instance. So we look at classifiers, and one problem there is that you have there is not really a reference, right? So the Human Cell Atlas is doing a great job into getting an atlas of cell types across the human body, and there is a specific immune atlas. But at the moment, there is actually no clear idea on what this atlas will look like. Who should we profile? Should we include people from different genetic backgrounds? Will it only be on healthy populations? But if we are classifying, for instance, samples that have been challenged with specific pathogens, how would the classifiers perform? There is another problem about labels. So I think for PBMC data, that's relatively easy, but for other data types, it's very, very difficult, like brain cell types, for instance. I mean, you have these hundreds of cell types that are identified by the Allen Institute, and then the same from Sten Linnerson's lab in Karolinska. So how do you match these cell types? Like, what is the identifier? What's the label that you use? And the last point, I will not touch upon this today, about sharing. So a lot of data is very difficult to share, actually, due to privacy issues. One very interesting approach over there is SC Arches from Fabian Theis's lab, where he developed this federated learning approach. So you actually don't have to share the data, you just need to share the trained model and then update the parameters of the model. In this case, it's a deep learning model, so you need to update the weights based on a new data set added at a different location. So this is very interesting. I'm not covering this today, but do have a look at their preprint if you're interested. Okay, so what we try to do is to solve this, these first issues of the fact that the reference is actually being built at the moment and that we have inconsistent labels across different data sets. So we came up with this hierarchical progressive learning approach. So I'll try to explain this briefly. There's a preprint again about this, so feel free to check it. But the basic idea is like this. So we have one data set where you have 3 labels, 3 cell populations. So they are notated 1, 2, 3. You have another data set where you have A, B, C, and D. And clearly from this plot, you would say that A and B are subpopulations of 1. And that's basically what we want to automatically learn or infer from the data without manually linking these populations together. So we want to come up with this tree basically at the end, where from a root we have 3 populations and one of them has 2 subpopulations. The way we do this is that we train a flat classifier on dataset 1. So just a classifier that is tasked with separating 1, 2, and 3. And we do the same for dataset 2. So we need to separate A, B, C, and D. And then we basically use this flat classifier trained on dataset 1 to classify cells from dataset 2 and vice versa. Right? And that what this will give us is that basically A and B will be labeled as 1 in this case. And part of 1 will be labeled as A, part of 1, another part of 1 will be labeled as B. And using this matching information, we basically come up with this tree automatically. Yeah, I hope this idea is clear. So to show you how this works, so we here have an example where we basically have 3 data sets, again, PBMC data. And these data are annotated at different levels. So for instance, if you look at CD19 positive B cells, you see that this cell population is present in all the 3 data sets that we include. While, for instance, if we look at monocytes, they are included as monocytes in batch 2, while they are included as 2 subpopulations of monocytes in batch 1, and only one of the 2 subpopulations is present in batch 3. Yeah. Eventually, this is the tree that we want to identify at the end, that from the root you go to monocytes, and then you subdivide monocytes into different subpopulations. So if we apply our approach to these 3 data sets, this is the tree that we learn at the end. So of course, some things are easy. As I said, B cells are present in all, so it's easy to define them as one population. Again, natural killer cells with their 2 subpopulations and so on. But there are also a lot of mistakes in this tree. So here again, the natural killer cells from one of the data sets is actually predicted to be a subpopulation of CD8 T cells, which is clearly a mistake. Here, for instance, CD8 naive T cells are grouped under the CD4 T cells, while they should be under the CD8s. This is a common problem. I think most classifiers actually make this mistake. It's very difficult to resolve the difference between these cells based on the mRNA data without looking at protein markers. Some things we're not really sure if they are mistakes or not. So here, for instance, you have these MK cells, so megakaryocytes, and they are the ones that come from one of the data sets, the EQTL study, are basically put as a subpopulation of the ones from the benchmark data. If you look at the UMAP, it does show you here that they are grouped together, they are in one cluster, but they are clearly separable. Like you can see the different colors over here. So it can be that they are slightly different population. So this would then not be a mistake. This would actually be that there is an overarching population and there is a subgroup of that population that is defined over here. And whether that's biologically relevant or not, I really don't know. Now, once we have constructed this tree, we can then use it to classify a new data set. So this is kind of the practical application of this classifier. So here we take a 4th data set that was not used in the training or in the building of the tree, and then we try to classify the cells in this data set. So the confusion matrix on the right here is basically showing you the true labels on the rows, and these are the predicted labels. So you see we have a much deeper annotation level from the tree that comes from the fact that the data used in building the tree was annotated at a deeper level. And in general, you see this classifier works quite okay. Some mistakes over here between the CD16 plus monocytes and mixing them with the MDCs. But in general, it's working quite okay. So to summarize what I told you today, cell identification from single-cell RNA-seq studies is moving from unsupervised approaches, which rely on clustering, visualization, overlaying markers, to more supervised learning schemes, where we rely on automated classifiers to do the task for us. And I think this is driven mainly by the increasing amount of data and the fact that we're doing a lot more samples. It was very common that you have, one sample coming from a few individuals in a paper, but now you see papers with hundreds and hundreds of individuals and millions of cells also included. So there is a lot of potential actually for these automated approaches over there. We did a comprehensive benchmark. We think this will help users to choose which method actually they are interested in. So if you're interested in a very deeply annotated data with many rare populations, then probably using a simpler classifier is better than a deep learning method, for instance. But also for developers, right? So if you're interested in developing a new classifier or whatever, I hope we give enough tools for people to make it a bit easier. And lastly, I showed you this continuous learning approach that will allow us to deal with this current limitation that we don't have a good atlas of PBMCs, for instance, to deal with. And with that, I would like to thank you for listening. All the work I presented today is the work from 2 PhDs in the lab, Tamim Abdelal and Leiden University. And yeah, thank you. I'll be happy to take questions.
Thank you very much, Ahmed, for sharing that. So as people think about what they'd like to ask, and like I said, I think it's a small enough group, so folks can go ahead and raise their hand and ask directly if they feel comfortable, or if not, drop the questions directly into the chat. Just to kind of get the conversation started, what I was wondering about is, as I was listening to that, you mentioned a series of easy problems, and I was trying to think about what are the hard problems in single-cell RNA-seq classification. And actually, I was glad that when you came to your last example, you highlighted that CD8 cytotoxic lymphocyte versus natural killer cell misclassification, which, for me is really one of the common bars, right? Can something deal with this kind of common issue that a lot of us run into? So I guess, as you've gone through the different comparisons and whatnot, have you found a particular method that can actually resolve those challenging problems like the CD8 versus NK without then you kind of manually going back in and trying to reannotate some of that? Or what do you think needs to be improved in order to fix that?
I think that's a very good point. So let me also point out to one more thing. So there is a lot of, so it is very domain specific, right? So if you're interested in PBMC data, I think you have, you're a bit more lucky than if you're interested in a system where there's not a lot of data. Because for PBMCs, a lot of people are actually looking into it. The same goes for the brain and lung data, for instance, pancreas. So these systems, I think, are very popular in single cell studies. What is very helpful in PBMCs is that you can also have these surface markers. And now with site-seq data, where you can combine barcoded antibodies together with the mRNA measurements, you can actually get a better view of the cells. I think last month or so, Rahul Satija and the Sierra developers released this very nice atlas, actually, that includes, I think, 250,000 cells, where they have mRNA measurements, but also 240 antibodies. And it gives you like a very detailed map that you will never get if you only look at the mRNA profile. So these multimodal measurements are very rich. I think they also showed that, for instance, if you come only with an mRNA data and then you use their reference and method to classify your cells, that you actually get a nice split, even if you don't have these surface markers measurements yourself. So that's, I think, is a very good approach and has very good potential. One limitation still there is that they rely only on 8 healthy individuals. So again, if your sample has diseased cells in it, it becomes really tricky
Yeah, we've found in some of our own work that disease really throws a wrench into everything, right? Because suddenly you have, basically the acquisition of so-called gene modules or cell states, right? That might start to muddy the waters in how some of these classifiers are learning if you're only feeding in that healthy data, right? As far as how to train your classifier. Let me see. Leon, yeah, go ahead.
Yeah, in that sense, that's why I'm really excited about this growing classifiers, or the ability to continue to learn, right? Because I don't think it's possible at some point that we say, we have seen all the possible cells in that system. That's very unlikely.
Yeah, and I guess in the kind of second part that you shared about the differences when you introduce the differences in platforms, right, and how the platforms might throw some particular challenges. What would your advice be to a group? Say they're halfway through collecting a cohort, and 10x releases V4, right, and they promise now double the genes and double the cells. How might you advise them? Do you suggest, stick with your current platform or go to something else? And we might have lost you temporarily.
Yeah,
you're back now.
My connection was dropped for a second. Sorry.
Yeah, you're okay. I was just asking, if you're advising a group and say, 10x releases a new method that is going to drastically increase gene capture, do you go into that, or do you stick with your kind of tried and true method, right? Do you think that some of the different classifiers or ways of integrating data are at the point where they can deal with different technical covariates, or would you say, it's better to expand your biological replicates and minimize that technical covariation, even though we can, correct for it or adjust for it computationally?
It's always a difficult question, of course.
Yeah.
I think the good news is that, at least for 10x, from what we have seen so far, differences are actually not that bad. I mean, there are clear differences. So if you integrate your data that was collected with V2 compared to V3, you would see differences in a joint embedding of the data for sure. But it's not super difficult to correct for, and at least in terms of classification from the one experiment I showed you today, it does seem that the best correspondence is still between the two chemistry versions. So that tells me that, okay, perhaps you should go with a newer one. Also gives you, as you said, a higher capture, and then you see a clearer picture in that sense.
Yeah.
But we still don't know. Huh? So one thing I really don't know yet, and we're trying to get a picture of that, is if you use, for instance, 3' versus 5' capture kits, how is this classification going to work? Because they do have different detections, right? There are very clear populations, like, I don't know, gamma delta T cells you can capture very well with the 5'.
Yeah.
It's very tricky on the 3'.
Yeah.
Yeah. So
actually, so now that you mentioned that, we should, I'll write to you after, because we actually have some 5' and 3' data off of the same samples that would be great to think a little bit more about. Yeah.
Yeah, that would be really nice. We have some data from Jimmy from UCSF,
yes.
which we will look into. So we'll try to see, how well can we classify these 5' cells without including them in the training set. But yeah, we'll be happy to talk, of course.
Yeah, that would be great. Does anyone have any questions, particularly any trainees in the audience who have any questions? Feel free to go ahead and ask. I think, Ahmed has really given us a tremendous view and really an unbiased view of different techniques and classifiers that exist. So I'd encourage anyone who's interested to go ahead and ask anything.
I'll ask a more philosophical question. I'm Sam. I'm one of Jose's trainees, so he set me up for this. So I mean, with the growing number of classification methods, and sort of the single cell computational field is quite crowded, I would say, these days. What do you think is, for a typical researcher, what's the path forward, right? Do they turn to, the methods like you've published to sort of identify the right way to use their data, or, how do we sort of democratize this process?
While you think about that answer, I'll correct it. I'm actually one of Sam's trainees. It's the other way around, but.
Okay. It is a very crowded field. On the other hand, There are a lot of challenges. So practically speaking, these classifiers are still difficult to use. I might have sold it well to you, but still, if you are really doing this yourself, you would see that these classifiers actually don't work that well. I'm really excited about the new Azimuth classifier from Seurat because I think it's a simple idea. It does work quite well. Again, it's not perfect. So you again have a very nice atlas that I think this is the best training data that you would get. But if you would try it on, I don't know, on the tumor immune atlas that was released from Holger Hein also a couple of months ago, it just doesn't work. So it's very challenging. How can we have a lot of data. If you look at the number of cells that are profiled every day, it's really growing like crazy. We don't have a way to put this all together to give us a good picture. That's not what's happening. Of particular interest, I think within the classification task, this rejection is really, really difficult to work with. So how can you reliably identify cells that have not been encountered during training I think is a very interesting approach. In the hierarchical progressive learning, we tried these one-class classifiers because we thought, if you train like a very tight boundary about what you know, it should be easy to reject what you don't know. It's still not the case. And I can also point you in the preprint to cases where this completely failed. So yeah, so I think focusing on the practicality of things is important. So even if it's a very simple approach using one of the already developed methods, just making it useful for people who do, I don't know, brain RNA-seq is something that we don't have at the moment.
Yeah, no, I would definitely agree with that. I mean, the usability, you clearly see methods that take off based on the usability, right? And hopefully they're also reasonable at what they're doing. Thanks, Sam, for the question. Are there any other questions as well?
I might have one question, a general question, because we also have several single cell RNA-seq projects, and I would like to get your suggestion about especially the project which, for example, have limited markers, and also limited RNA-seq database which we can use to do this kind of cell class annotation. So for this type of project, do you have any suggestion, like which database, public database we can go to find the well-known markers, and also what type of methods we can use to do the cell cluster annotation? I mean, this is a very general question, but I think it's also a challenge for us right now. Do you have any suggestions?
To make sure I understand correctly, you mean you're profiling a tissue where you don't have a lot of previous data? And is that what you're saying?
Right.
Yeah.
So for example, this tissue actually, we know that there's some maybe some de novo cell type still, but we don't know that yet. And then we really have limited marker for that.
At least from my experience, at least the biologists sometimes have an idea, at least about what to expect at a larger scale, right? But when it comes to like finer identification of subpopulations of cells or substates, I think if you really don't know, then relying on differentially expressed genes or markers that you can infer from the data itself, and then later validating those with an independent approach with stainings or whatever. I think that's the best I know at the moment.
So yeah, I know this kind of weird question was but sometimes if we just say basically so for example single R, we actually have RNA-seq database, public RNA-seq database, and also some people actually also use microarray database to do this kind of machine learning method or the correlation analysis to identify the cell cluster. But if we don't have any well-known markers, which actually show up in the differential expressed genes, then researchers maybe also get a kind of they don't have a very high confidence to say, okay, this is the clusters, because there's a very high correlation values with this cell class in the RNA-seq database. And then,
but then doesn't that question your definition also of that population as an independent or like a cell type? I mean, this marker identification differential expression based on clusters is already very inflated in terms of p-values, right? Because we use the same data to cluster, and then we use the same data to find markers, which is, we shouldn't do that, but we do it. But then if you don't find markers, then I would really think maybe you actually don't have a distinct population in that case. It's very unlikely that you don't find markers. Perhaps then you're looking at a process that is going gradually from like a differentiation or something. And in that case, I don't think classifiers are actually the best approach to go. So you would rather do some sort of pseudo-time inference and then alignment of the trajectory to a reference trajectory or something.
Yeah.
Okay.
All right. Well, thank you very much.
I have a question if the time allows.
There's one more question. Okay, yeah, go ahead.
I'm Walid. I'm from Brigham and Women's. Thanks, Ahmed, for the nice presentation. So I have 2 quick points. The first one is regarding the hierarchical learning. So you showed a UMAP representation. I was just wondering, before you follow up and do the downstream analysis for the hierarchical learning to make sure the annotation are consistent, did you try to calculate an embedding like a UMAP of the 2 data sets with different annotations? I expect the annotation you show as 1 and on the other data set as A and B. I think they should end up compactly located in the embedding space, which might mean they are the same cell types. Am I correct in that? And did you try to do it?
So we didn't try that. I think they will overlap, yes. Yeah, I'm trying to think a little bit about what you say. So what you say is that we can also use the embedding to infer this hierarchy. So for instance, if I see that cluster 1 and 2 lie within cluster A, then we should infer this. Is that what you're saying?
Exactly.
Yeah, there is a bit of a problem because these embeddings are actually not very representative of what we have in the high-dimensional space. You do see it immediately with, for instance, the different subpopulations of CD4 cells and CD8 cells. Yeah, we do have this very clear idea on how these cells are hierarchically organized, but that's not what you see in a UMAP embedding. So I'm not sure if you can actually use it for that.
If they have the same expression in terms of high dimensional feature, if they are similar and there is no patch effect, of course, if you measure the pairwise similarity of data sets that should be annotated the same, I expect they should be located close to each other.
I think this will make our life easier, but unfortunately that isn't right.
Okay, the second point is, did you try looking at the deep learning approaches where basically you can overcome some of the challenges you mentioned about large scale analysis, and especially with deep learning, we have experience applying it on data with the feature space was really huge. So the scalability on feature space and scalability on number of data points, in your case will be number of cells, were very, very promising. Did you try to also include it in your analysis as well?
Yeah, so in the benchmark, as I showed, there are multiple of deep learning approaches over there. I think I really want to make it clear that the message is for a classification task, these deep learning approaches, I don't think they are necessary because the problem is easier than you think, actually. At some level. So the problem is not with the nonlinearity, let's put it this way. But these approaches are very important and useful for other tasks, right? So I am specifically interested in these variational autoencoders for learning joint embedding spaces, for instance, or trying to predict the effects of perturbation or stimulation on cells, which, yeah, I think that they will be very, very handy in these settings. So that's a bit my view, but for the specific task of classification, I don't think they are useful.
All right. Well, thank you very much for that, and also spending a little bit more time to answer all of our questions. I think it's probably dinner time for you over from where you're calling from. So appreciate you hanging out, and thank you so much for taking the time. Very much appreciate it.
Thanks for the invite. Quite enjoyed it. And do let me know about the 5', 3'.
Yes, yeah, what we'll do. Take care.
Cheers.
All right.
Bye.
Thank you. Bye.